Okay. Good afternoon, everyone. My name is Rizwan. I'm sitting here in Hyderabad office, Share India. So I'll be taking the second session for the day, which is sample collection and management at ART Center. The main objectives of this session are what is pre-sample collection process, and then how do you process the sample? What is the collection process? How do you allocate it, transport it, and what is the acceptance and rejection criteria for HIV viral load 1 testing? And then if there is any discrepancy, how do you manage the discrepancy? So the routine flow of patient is when the patient comes, they get registered, they see the medical officer, and then the medical officer requests for a test, if either be CD4 or viral load. Then the patient goes to the lab or phlebotomist wherever the blood is collected, uh, and blood has to be collected at that place, and then simultaneously entry has to be made into IIMS, Integrated Information Management System, which is nothing but your SOCH, day-to-day activity which is being used as SOCH. And then the collected blood has to be centrifuged, plasma has to be separated, stored in the refrigerator, and transported to the viral load lab with the cold chain, that is within the temperature range of 2 to 8 degrees. Now the temperature and the time frame are a little bit trickier, and very stringently this has to be followed. We will talk about it in the subsequent slides. Pre-sample collection process. As soon as the patient comes, actually, I wanted to make this as an interactive session, but since there are so many participants, I will assume everyone knows this, but this is the practice. Okay. As soon as the patient comes, you are supposed to cross-verify their green book against the white card and check for whatever test has been requested. Either it's a CD4 test or a viral load test. Based on what test has to be done, or both the tests have to be done, you have to select your vacutainers. We are using only EDTA vacutainers, which are lavender color caps. Yeah, either for CD4 and for viral load. For both the tests, we are using the same vacutainers. For CD4, you require 3 ml of blood, and for viral load test as well, you will require 3 ml of blood. If you only have to collect CD4 samples, just draw 3 ml blood. If it is for both CD4 as well as viral load, using the multi-gauge needle and the holders, insert the needle once, and you can change the tube twice. Collect 3 ml of blood in 2 different tubes. So the 3 ml of blood is required, minimum volume, because that gives you a plasma of about 1 to 1.5 ml, depending from, it differs from patient to patient, as well as the volume you have drawn. So 3 ml is required, as it gives you at least minimum of 1 ml of plasma. That is a requirement. That is the minimum required value, volume you have to send to the viral load lab. Okay. And then a 17-digit barcode is provided to each identifier number. UID is provided to each individual for particular test. Let me just highlight this around. Okay. So here we have an example where it says ART. Now across India, for all the patients, everyone, the ART will remain the same. Followed by the next 2 digits will be the state code. MH stands for Maharashtra. Similarly, TS will stand for Telangana, and every state will have their own 2-digit code. And then the next 3 digits, the next 3 alphabets will be the district in that particular state. And then the next 2 digits, the 01 over here, will be the center code within that district. And followed by the 5-digit patient ID, which is unique to that particular patient within that particular ART center. And this 02 represents the number of tests, viral load tests that this is. Let's say patient has already gone 3 viral load tests. Now the patient will have in the end, the number will become 04. So that number keeps on changing with each and every test. The first viral load test will be 01, the second will be 02, the third will be 03, and subsequently. Ensure that you label your vacutainers before drawing the blood, because this might lead to some transcriptional errors at the end if you have not labeled them beforehand. Okay, whole blood should be collected with a single prick. 3 ml EDTA tube should be used, as this will give you at least 1 or 1.5 ml of plasma aliquot after aliquoting. Details of sample collection should be recorded in viral load lab register, as well as they have to be entered in SOCH. I hope all the ART centers are maintaining the viral load lab register. If you have not maintained it, please make sure you maintain these viral load lab registers, and you make entries of each and every day on that particular day, as well as do SOCH entries. There should be a sample collection manual available at each sample collection site. So these are some of the prerequisites, some of the supplies that you will need. Here, the most important thing is the powder-free gloves. The more emphasis on powder-free. The reason being, if there is any powder in the residue or the blood, this will hinder with your PCR process, and you will not get a report from the lab. Then you will need EDTA, vacuum cleaners, needles, holders, tourniquets, alcohol gauze, cotton gauze, alcohol, sanitizer, your viral load lab register, as well as a puncture-proof container. This practice has been seen at some of the places, and we strongly recommend you ensure that you have a puncture-proof containers. Some of the facilities, what we have seen, they were not using, and it is for the safety of the technician and for the biomedical waste management people, whoever are using, make sure you have puncture-proof containers. Okay. These are a few of the steps of sample collection. First, the safety of the technician or the person drawing the blood is much important. So ensure you have right PPE, you're wearing correct gloves, being powder-free gloves, and your apron, mask, or face shield, whatever is required, or whatever COVID guidelines you are following. Locate the vein, clean the area, let the area dry, and then draw the blood with the same prick, single prick. And the important point in this slide is you have to ensure that you invert this tube, EDTA tube, after drawing the blood 8 to 10 times. This is very important. Now, what happens if you do not invert it is you'll get clots and the blood will get clotted. You will not draw a plasma. And there is another possibility. You just shake it or did not mix it properly, did not invert it 8 to 10 times. That will lead to microclots. Microclots are not visible to our eye. When we put it in the machine detects the clots and the machine will not give you the result. So viral load lab will discard the sample. They will ask you for a repeat sample. And the whole blood that is drawn has to be, there are two key points. The whole blood that is drawn has to be separated within 6 hours. So plasma processing should happen within 6 hours. If it does not happen, the RNA stability is lost, and you are most likely to get T&A reports out of it, false T&A reports. And then heparin tubes must not be used, as heparin is a PCR inhibitor. And these blood samples are to be kept at only 15 to 30 degrees, that is room temperature. Some of the regions which have hotter climates during summer, which reaching around 40 degrees, 45 degrees, ensure as soon as you have drawn the blood, immediately centrifuge, separate the plasma, and store the plasma in the refrigerator, that is 2 to 8 degrees. Make sure you do not leave any sample more than 6 hours, that is whole blood sample. So within 6 hours, the whole blood has to be centrifuged and the plasma has to be separated. Okay, now we have a quiz. So the poll will pop up. I request everyone to answer this within 30 seconds, please. The question is, powder-free gloves should be used by ART lab technician while HIV-1 viral load sample collection. Is this statement true or false? So I would again emphasize on this point that powder-free gloves should be used for viral load testing. If you use powder-free gloves, the reason why we are asking you have to use viral load, you have to use powder-free gloves is, if you use powdered gloves, and if that powder goes into your blood sample, it mixes and it stays in the plasma, and that plasma will go to the machine in the lab, and in the lab while testing, the machine will tell you that it will not give you a correct result, it will not generate a report, it will give an error code. So only powder-free gloves should be used. Powder inhibits PCR, and the mechanism what we are testing is with PCR. We are detecting RNA copies. So please ensure that you have powder-free gloves in your ART center. Collection day and timings. Ideally, it is collected from 9 a.m. to 1 p.m. from Monday to Saturday, except for public holidays. However, varies from place to place based on the norms. Whatever norms you are following, whatever are your timings, please you collect the sample at the same time. Another important point I would like to add here is if the patient comes after 1 o'clock, please do not send back any patient. Draw the blood sample, arrange for the transportation, or store the blood in the refrigerator. The plasma, sorry, not the blood. So the plasma can be stored in the refrigerator, that is 2 to 8 degrees, not the freezer compartment, the fridge compartment. There you can store the blood, the plasma sample for 5 days. Change in collection is subjected to prior approvals from in charge, as well as informing the transport agency. So the storage, yeah, there is again emphasis on the same point that the whole blood should be separated within 6 hours. 6 hours is the time here. Within 6 hours, if you have not separated the whole blood, that will lead to RNA degradation, and it will give you a false result. It will generate a copy number, or it will generate a TND, but that cannot be relied upon, because the stability of RNA will be lost after 6 hours, 6 hours of blood collection. And centrifugation has to be done at 2000 to 2500 RPM for 10 to 15 minutes. Please do not do it only for 5 minutes, as it might lead to microclot formation or improper separation, or unclear buffy plasma generation. Ensure that you have a very clear plasma. Use either pipettes. If you have pipettes, ensure that you are using aerosol barrier tips. If you do not have pipettes, make sure you are using single wrapped pasture pipettes. The pasture pipette is similar to a dropper. You will get it in either bulk or single wrap. Ensure you have single wrap and sterile. This avoids any contamination or any dust, or maintains the stability as well. After drawing the blood, after centrifugation, whatever volume of plasma you get, be it 1 ml, 1.5 ml, or more than 1.5 ml, please transfer all the plasma into a separate 2 ml cryotube and dispatch it to the viral load lab. If, let's say, in we suggest to transport as much of plasma you draw, because in viral load labs, sometimes the test might fail. There is some issue with the machine. There might be some issue with the machine. Because of the reason, they will have to repeat the test. If you send extra volume, whatever you have drawn will be stored. The viral load lab stores the blood sample for 4 months. So they can repeat the test, and once the report comes, they can immediately give you the result. Otherwise, if they don't have enough number of sample for second run, if something happens in the first run, then they will inform the ART center, and ART center has to call back the patient, redraw the sample, and again send it to the viral load lab. Okay, and then transportation will be in 2 ml cryotubes. There is a change in the policy now, irrespective of either the co-located or non-co-located. This point has already been emphasized by Mr. Pankaj, that everyone is supposed to separate plasma and send. Previously, the practice was most of the co-located centers were sending the whole blood to the viral load lab, but according to the latest norms and latest policies, everyone, all the ART centers, either co-located or non-co-located, have to separate the plasma, process the blood, and send only plasma to the viral load labs. No one is supposed to send whole blood. So once the plasma is separated, it can be stored in the refrigerator 2 to 8 degrees for a maximum period of 5 days. So there are two different time frames here which has to be remembered. The first time frame is when you draw a whole blood sample, you only have 6 hours of time to process the sample and generate separate plasma. Once the plasma is separated, that plasma can be stored in 2 to 8 degrees. That is a normal refrigerator for 5 days, not more than 5 days. If it is stored in 2 to 8 degrees for more than 5 days, again the RNA will degrade. So these are the two important time frames which has to be remembered. Whole blood has to be processed within 6 hours, and plasma should be stored in 2 to 8 degrees for not more than 5 days. And you have to make sure that your blood samples, whatever you have drawn, are processed within 6 hours, stored in the refrigerator, and within 5 days they reach the viral load lab. And repeated freeze thawing should be avoided. This is not freezing, but still, putting the samples in the refrigerator and again bringing it out and again transferring it into a 2 to 8 degrees container. This has to be avoided. Make sure the stable continuous 2 to 8 degrees of temperature is being maintained. So there's another quiz here that has popped up. So the question is, plasma must be separated within 5 days, 24 hours, 6 hours, or all of the above. I request everyone to please answer the question as soon as possible within 30 seconds. Two of them have said it is 24 hours, 130, the majority has said it is 6 hours, and someone has said all of them. So this is an again emphasis for your clarification, for better understanding, and for following the process correctly. The plasma, the blood when you draw, after the blood is drawn, within 6 hours, the correct answer is 6 hours. Within 6 hours, you have to separate the plasma. Previously, in the previous slide, I have said there are two different time frames what have to be considered. That is, when you draw blood, within 6 hours, the plasma has to be separated. Once this plasma is separated, this is the second time frame. When the plasma is separated, after that, you can store it in the refrigerator, that is 2 to 8 degrees for 5 days. So I hope this clarifies any doubts anyone had. And I request participants to please do not draw on the slide. Okay, so sample packaging, schedule packaging, and dispatch. Here, what we'll see is ensure that the sample aliquot reaches the viral load lab on or before 5th day of sample collection. As the ART centers do not have a -20 or -80 degrees, you can only store it in 2 to 8 degrees. Once you store it in 2 to 8 degrees, the transportation will also be in the same conditions. And viral load labs are equipped with the machines, are equipped with the freezers to store the sample for more than 1 year. So you have to ensure that all the samples that have been drawn are processed within 6 hours, stored in 2 to 8 degrees, and reaches the viral load lab on or before the 5th day of sample collection. Collocated and non-collocated both have to centrifuge the blood samples, separate the plasma, and send the plasma only, not the whole blood. Preparing for shipment, sample transportation sheet is required. Box containing sample for aliquots is also required. And then you have to verify the samples against the transportation log, whatever you are maintaining. The correct number of samples are entered and are sent. What are the prerequisites for packaging? For prerequisites, if the transport agency is not bringing their own vaccine carrier or their own 2 to 8 degrees box, the ART center is supposed to ensure that the box is ready before the transportation agency comes for the pickup. So we need is the overnight frozen gel packs. The gel packs, the frozeable packs, they have to be kept in the freezer, not in the fridge compartment, in the freezer compartment where it can be frozen and ice can be formed. It has to be frozen for overnight. That is in the whole night. Almost 12 hours is required for the proper storage for this to reach that correct freezing temperature, freezing conditions, which will cater to 2 to 8 degrees during the transit. Ensure all the plasma samples are labeled, sealed with the paraffin. Aliquot storage boxes available, sample transportation sheet, sealable plastic bags. Sealable plastic bags are recommended because when you send the TRFs along with the blood sample are also supposed to be sent. So the TRF can be placed in a sealable plastic bag, sealed, so that the water or the leakage will not destroy the TRF. Absorbent pads or brown paper. This is important. I'll show in the next slide why you need absorbent pads. And then clean transportation box with a biohazard symbol and label and name and details of the sender and the receiver. That is the ART center and as wherever we are sending it to respective viral load labs. The packaging is triple layer of packaging, wherein the primary layer is when you separate the blood and plasma, which is kept in a 2 ml cryo box that becomes the 2 ml cryo vessel. That cryo tube becomes your primary packaging, and you pull how many samples you have done, either 10, 20, or put those X number of tubes in a sealable plastic bag. This becomes your secondary packaging, and then the sealed plastic bag is supposed to be kept in a vaccine carrier or a cold chain box, which can maintain 2 to 8 degrees temperature. So this is how, if you do not have a vaccine carrier, if then this is the procedure to ensure that the cold chain box is prepared. First, overnight frozen gel packs are to be placed at the bottom. Over it, place absorbent pad or cotton or tissue paper. If you do not have any of this, you can also put some newspaper over there. The principle for this, the reason for putting this is the gel pack will leak some of the water that can be absorbed by the gel, this absorbent material, as well as in case if some samples have tubes have broken or there is a leakage of sample, then this will absorb everything and prevent the contamination to the environment. Biohazard symbol is also important, and ensure that all the TRFs and required transportation sheets and other documents are sealed in a Ziploc cover and sent to the viral load lab. On the day of sample collection, confirm that time of sample pickup. Just with your sample transport agency, ensure what time they are coming at. Whenever, at whatever time you have your scheduled pickup, ensure that make sure that your transport box, just the box without sample, is ready half an hour before. The reason for it is it will take time to cool down the box. So you just place all your gel packs, put absorbent material, close the box, leave it for half an hour to 40 minutes. After that time, only then you can transfer your samples from the refrigerator to the cold chain box, because the cold chain box is at the room temperature. It has to attain that 2 to 8 degrees temperature. Then you transport the samples in a cold chain that is maintained 2 to 8 degrees. Ensure you are recording the temperature, number of samples that you are sending in each day. Hand it over to the transport agency and tell them this is a biological material, a biohazard substance. Do not shake or keep it in the upright position. Do not tilt it or throw it upside down. And you have to do IIMS SOCH entries within 48 hours of time, as SOCH does not give you any further time for entries. Discrepancy management. Okay, for that, this is the acceptance as well as rejection criteria of samples. The acceptance criteria is sample should be properly labeled. The sample tube integrity should be maintained. That is, it should not be leaked or broken. Sample label matches the TRF, and adequate volume of sample is present. A clear plasma is there, and the transportation happens within 2 to 8 degrees of temperature, because when the viral load lab receives the samples every day, they check the temperature out and what conditions it has been received. If it is received on temperature, let's say 15 degrees or 20 degrees, they reject the samples and inform the ART centers and request the repeat samples. And the rejection criteria is either hemolyzed samples or crossly lipemic samples, contaminated samples, leak tubes, inadequate volume, improper labeled samples, temperature deviation that is more than 8 degrees, or samples from HIV-2 infected individuals. Here in our national program, we are only testing for individuals with the viral load. We are only performing viral load test for HIV-1 and 2 co-infections. We are not doing any viral load for HIV-2 infected individuals. So discrepancy will be informed in any case, either rejected or an error or a request of sample, repeat of sample. The viral load lab will inform the ART center over the phone as well as with the email, and ART support are expected to resolve this discrepancy within 48 hours or address it by the same within the same time frame. If a particular sample is rejected for X reason, you have to mention the reason. The viral load lab will inform the reason of sample rejection. The ART center are expected to against the remarks column in the viral load lab register, enter the reason for rejection of the sample and call back the patient and draw the blood again. Here are some of here we have given some of the examples of the reasons why rejection discrepancies can happen. Let's say a total number of samples allocated not received as per the transportation sheet. You have mentioned in your transportation sheet that 50 samples have been drawn, but the viral load lab is have received only 45. Five of the samples are missing. So this could be one of the discrepancy. And then sample aliquot received but not updated in IIMS, that is SOJ. Uh, that is what I was emphasizing. The SOJ entries have to happen on the same day or immediately the next day, because the SOJ does not give you a trace back time of more than 2 days. Slide. Okay. Biosafety and biomedical waste management. So every ART center has a designated workspace for sample collection and liquidating. Ensure safety of staff and healthcare providers. You also have to ensure the safety of the staff handling your biomedical waste. Those people are usually unaware of the samples and the impact it can have on their health. So ensure you train your cleaning staff and housekeeping staff on how to deal with how to dispose of the biomedical waste, how to transport it. Make sure you have enough PPE on you, that is your gloves, mask, face shield, apron, or whatever it be. Hand wash. There's a beautifully addressed here. Always hands your wash before and after removing gloves. And as gloves are not a substitute for hand wash. God has taught us this thing very clearly, that how important hand wash is, especially in the healthcare sector where we are dealing with infectious diseases. This plays a crucial role. So make sure hand hygiene is followed. Decontaminate your workspace and process the spill according to the spill management criteria. There are three kinds of bins or color coded different things. The yellow bags are for cotton swabs. The red bags are for gloves, disposable pipettes, or pipette tips or vacuum cleaners with leftover blood. And the white box, that is puncture proof box, is only for needles. Here, I would request one of the participants or anyone can please unmute themselves and answer this question. In the puncture proof container, is there a requirement of hypochlorite solution? 1% hypochlorite solution. Is it required? Is it required to pour 1 ml, 1% of sodium hypochlorite preparation, sodium hypochlorite every day, or you can directly discard the needles in the puncture proof container without hypochlorite? I request someone to respond to this question, please. Okay, any of the ART centers lab technicians question, do not put hypochlorite in your puncture proof containers. The latest biomedical waste guidelines says not to use any hypochlorite solution. Just like that, put your used needles in the puncture proof containers. Once it is 3/4 filled, seal it and give it to the biomedical waste management agency. Okay, the discussion points is how do you avoid stock outages? Always you have to ensure you have enough number of stock. Let's say ART Center X is expected to send 50 samples in one day, and that same day we have seen there is no vacuum cleaners. What can be done? If there is, we can either borrow it from someone else. But why did this happen? Because the inventory was not managed. So ensure you manage your inventory, plan it accordingly, plan everything accordingly, and transport the samples on the same day. How will you schedule a sample transportation? This will be done through SACS. SACS will hire a transport agency, and they will inform you of the dates and days of sample collection. On those particular days, you have to transport. Entries into SOCH have to be done on the same day, or it can be done on the next day. You only have 48 hours of time for doing SOCH entries. And then sample rejection. Let's say there's a sample rejection in the lab because of microclots. The lab will inform ART center that this particular sample has been rejected because of the microclots. In this case, the ART center's responsibility is to call back the patient, draw the sample again, centrifuge it, separate the plasma, send the plasma to the viral load lab, and make the entry in the occurrence register that this particular sample has been rejected for this reason, and this was the follow-up activity that has been done. So thank you everyone. Now the floor is open for questions. If anyone have any questions, they can please ask or put it in the chat box. Okay, I see one question. Can plasma be stored in deep freezer in temperature between 2 to 8 degrees temperature before transportation? So you can store it, but it is not advised because the transportation will be at 2 to 8 degrees. So it is advised that ART centers store the blood at 2 to 8 degrees itself. If you store it in deep freezer and then the transportation is at 2 to 8 degrees, that leads you to a freeze thaw cycle. So please avoid this. It is recommended that you store it in the 2 to 8 degrees itself. Can we do viral load jaundice sample? No, you cannot do viral load jaundice sample because the platform or the kits that are being used in the viral load lab is only for HIV-1 viral load testing.
Well, sir, just to inform, participants
Yes.
can unmute and also ask the questions.
There is a question, sir, why viral load is, what is this? Viral load not done for HIV-2 infections. Okay. The answer for this is why viral load is not done for HIV-2 infected individuals is there are multiple reasons. Some of them are the disease progression is very slow. There are very few number of HIV-2 infected cases, and the kit and the test what is done in the viral load laboratory that caters only to HIV-1 infection. It does not cater to HIV-2 infection. So the kit also does not supports the Abbott machine and the kit what we are using in our national program. Over to you, Pankaj.
So one question. Yeah, please,
Yes,
please.
please go ahead.
In the UID, if the sample is rejected and if we retest and send again, it contains of 17 numbers. So the
Yes.
last 2 digits represents the number of tests. So do we use the same number or it will use the next number?
Yeah, this is a very good question. Yes, you will change the number. Even if let's say you have drawn sample number 3, the last 2 digit was 03. The viral load lab, you have entered the same number in the source and then the sample got rejected. The viral load will inform you and they will reject the sample from their end. Then you have to enter it as 04. The number will change for the repeat samples as well.
Okay,
I hope
thank
that
you.
answers your question.
Yes, thank you.
Thank you.